be back.
I'll be back too. Okay.
Be back.
I'll be back.
Hello, everybody. We're going to start maybe 1 or 2 minutes late. We have a bunch of people coming in, and I am admitting them, or trying to. All right. Are you ready, Joanna?
Yes, and
Cool.
You can hear me and see my intro slide?
I can hear you and see your intro slide.
Then I'm ready.
Looks great. I'm very curious what you're going to say about that slide. Anyway, so hello, everybody. We're going to get started. Thank you, everybody, for showing up. Momoko, if you could, can you admit people as we get going? I'm not going to
Sure. Yeah, of
pay
course.
attention to all the people coming in.
Yeah.
But anyway, so thanks everybody for showing up. We're very glad to have you here today for our NextGen seminar on epilepsy. And today we have Joanna Mattis, and she is a epilepsy fellow at the University of Pennsylvania. And she's going to talk to us today about a mouse model of epilepsy, SCN1A mouse model of epilepsy, and a little bit about Joanna. So she did her MD, PhD at Stanford. And so she's a clinician scientist, worked with Carl Deiseroth's lab at Stanford, working on the hippocampal septal connections using optogenetics to dissect out that circuit. And she moved, was it in 2015, Joanna?
Yes.
Yeah, I got that right. So 2015, she moves to the University of Pennsylvania to do her residency, and she's been working with Ethan Goldberg on this Dravet syndrome mouse model of epilepsy, and we're very excited to hear her research, and so I will let you take it away.
Great. Thank you so much. And I'm really excited to have this opportunity to share my research with you. So thank you so much for this introduction. As was said, my name is Joanna, and I'll be sharing with you the work that I've been doing in Dr. Ethan Goldberg's lab at CHOP, investigating the corticohippocampal circuit in an SCN1A mouse model of epilepsy. And before I begin, I will spend a minute explaining the background to my intro slide. So this is, I think, a very beautiful painting by my friend and colleague, Dr. Emily Ferenczi. Emily and I were graduate students at the same time in Carl Deiseroth's lab. And she is also a physician scientist in neurology now in Boston. And I like to include this picture because, first of all, I think it's a very beautiful image. And it's nice to remember the sort of aesthetic beauty of the science that we do. And also because there are elements within this image that are sort of central to my research. So it sort of serves as a high-level conceptual background. So you can see on the top of the screen in the sort of aqua color, there's what could be a neuron that's being probed by a patch pipette. In the bottom part of the screen, that undulating structure could be a 7 transmembrane domain protein sitting on the cell membrane, transducing a signal into the cell. And then in the top right of the screen, that could be a sun or a light source. And light and the use of light as a research tool is a recurrent theme that I'll come back to over and over again in my talk. So as a physician scientist in epilepsy, a really central question that I think about a lot is how can we advance our field to better serve the around 1/3 of our patients who have medication refractory disease despite optimized medical management. And if we could better understand the mechanisms underlying epilepsy and seizures, it may open up new directions for development of therapy. And we can think about epilepsy mechanisms across a range of levels, sort of from the micro to macro scale. So at CHOP and elsewhere, there's been a lot of advances in understanding genetics of epilepsy. And there's also been a tremendous amount of work characterizing what can go wrong in brain with epilepsy at the level of the single cell. And then, as was mentioned, this year I'm a clinical epilepsy fellow, so I'm spending a lot of my time thinking about epilepsy and seizures at the level of resolution of a scalp EEG, which is multiple cubic centimeters of brain tissue or brain regions. But between the single cell and the entire brain region, there's a huge wide gap that I would argue is maybe our weakest spot of understanding of epilepsy mechanisms. And within this gap, bridging the micro and macro scale is the neuronal circuit. So circuits have been historically really difficult to study in neuroscience because of the way that the brain is organized. So as you all know, the brain is made up of many different types of cells that can have completely different roles within a circuit, and these cells can be sparsely interspersed and distributed within the brain tissue. So you can appreciate that if you wanted to study just the contribution of the red cells, for example, shown here, it is a non-trivial challenge to be able to specifically tweak just those cells within the brain. And I'll show you the same image again later in the talk, so you can keep your eye out for it. So with that kind of motivation, the overview of my talk is, first of all, to introduce two really wonderful, powerful tools we have in circuit neuroscience to study defined cells within a circuit. And then I'll also separately introduce the SCN1A mouse model of Dravet syndrome and the corticohippocampal circuit. I'll spend the majority of time in this talk sharing with you my data investigating this circuit within the SCN1A mouse. And then this is work that I've been doing, as I mentioned, with Dr. Ethan Goldberg through the NIH R25 funding mechanism as a neurology resident and fellow. And then I'll end the talk with just a few quick kind of high-level slides about possible future directions. So now I will introduce to you the idea of optogenetics, which is one of my favorite subjects to discuss. So shown here are two different bacteria, and bacteria around the globe have evolved within them light-sensitive ion pumps and channels that can be used to detect sunlight. So for example, so the bacteria can know where the sun is, and they can grow on the sunny side of the rock. These are light-sensitive ion pumps and channels that, in response to light, change their conformation and conduct ions across the membrane. And depending on what ion it is and which direction it moves across the membrane, this would result either in depolarization or hyperpolarization of the cell. And the amazing thing is that we can take these bacterial proteins and with just a few tweaks, express them within mammalian neurons in the brain. And this is an incredibly powerful research tool because with electrical stimulation, you have no way to distinguish between adjacent cells. If you wanted to study one cell or activate one cell with electrical stimulation, you would also recruit all of the nearby neighbors and fibers of passage. But with optogenetics, if you can engineer your system so that just the cells that you're interested in studying are expressing these proteins, then when you subject the tissue to light, you'll selectively manipulate only those cells of interest. And I had the great privilege, as was mentioned, of doing my PhD in Dr. Carl Deiserath's lab at Stanford. And so Carl, I think, is a scientific visionary and really the founder and pioneer of the field of optogenetics and neuroscience, which has really kind of revolutionized what we can do in circuit neuroscience. And just briefly, as a PhD student, my first major effort was to lead a kind of comprehensive characterization, optimization, and testing of the multiple excitatory and inhibitory optogenetic tools at that time. And actually, that painting I showed you on the first slide was the cover of the Nature Methods issue in which we published that work. And then I co-led a second methods effort to develop new strategies to selectively restrict expression of these optogenetic tools to just specific cell types of interest. And then finally, I collaborated with Dr. John Huguenard's lab at Stanford, an epilepsy researcher, to investigate this long-range inhibitory projection in the brain from hippocampus to medial septum. So switching gears now, I'm going to separately introduce a second technique in circuit neuroscience, calcium imaging. That is a really powerful way in which we can, on a large scale, monitor neuronal firing within defined neuron types. So GCAMP is a genetically encoded calcium indicator that fluoresces when it binds to calcium. So the image I'm showing here is an image I took in Dr. Goldberg's lab, and every one of those little donuts is a neuron expressing GCAMP. And this slice is relatively quiet at baseline. These neurons are mostly not firing action potentials, so the intracellular calcium levels are low and the GCAMP expression is dim. When I activate this slice, the cells fired, calcium levels go up, and I hope you can appreciate that for many of those cells, the GCAMP expression gets a lot brighter. So I think of optogenetics and calcium imaging sort of as two sides of the same coin. With optogenetics, we are controlling neuronal firing with light. So light causes ions to move across the membrane, which can trigger action potentials. Whereas with calcium imaging, we are observing endogenous neuronal firing with light. So spontaneous action potentials lead to an increase in intracellular calcium, which results in an increase in fluorescence. So it's a sort of optical dial in versus optical readout. And these two techniques are complementary to each other and can powerfully be combined within a given experiment. And I'll present one such experiment by the end of this talk. So with this kind of research background as a physician scientist, my goal in terms of addressing this challenge of advancing our field is to use preclinical animal models of epilepsy together with these molecular genetic techniques in circuit neuroscience to understand how and when neuronal circuits give rise to seizures. And what I hope to convince you of in this talk is that Dravet syndrome offers a really powerful tractable model to be able to link across scales of pathology in epilepsy. So Dravet syndrome is an often devastating developmental and epileptic encephalopathy, and the genetics of Dravet syndrome are relatively well understood. It arises from pathogenic variants in the SCN1A gene that encodes the NAV1.1 alpha subunit. So in our lab, we use an SCN1A haploinsufficient mouse lacking one copy of the gene and having decreased NAV1.1 expression levels. And there's been a lot of work to understand this mouse model at the single cell level. And with some nuances, the bulk of the data is points to inhibitory cell dysfunction as the major deficit in this mouse. And then at the whole animal level, these mice exhibit hyperthermic seizures. In other words, when you elevate their body temperature, they have seizures. And this is really interesting because human patients with Dravet syndrome have terrible seizures triggered by fevers. So it's a nice link to the human condition that we're modeling. These mice also have interictal discharges, SUDEP, and comorbid behaviors seen in patients with Dravet syndrome. But again, this intermediate level of understanding between the micro and macro scale on the question of what neuronal circuits are dysfunctional in these mice remains relatively poorly understood. So where in the brain should we look for a circuit deficit? There's converging evidence that points to the hippocampus as potentially a key locus for generation of hyperthermic seizures in these mice. So this is data showing intracranial recording from hippocampus and several cortical regions during the onset of hyperthermic seizures. And what they found was ictal activity was observed within hippocampal traces, and I've highlighted that with those yellow bars multiple seconds before it was detected within the cortical recording. This is data from a different but related mouse model showing really robust activation of the hippocampus following hyperthermic seizures. And then finally, a recent paper from the Catterall group used a mouse that instead of having decreased NAV1.1 expression throughout the entire brain, they were able to focally, just induce a focal lesion specifically within the hippocampus. So the entire brain was normal, except for a focal decrease in NAV1.1 expression restricted to hippocampus. And just that hippocampal NAV1.1 decrease was sufficient to confer the hyperthermic seizure phenotype in those mice, shown in red, compared to wild type in blue. So how could the hippocampus give rise to seizures? We thought about investigating the corticohippocampal circuit because of the way in which it's organized. So the entorhinal cortex sends a really strong excitatory projection onto the dentate gyrus, the entry point of the hippocampus. And then that signal is processed within dentate such that what is then propagated forward to downstream hippocampus is substantially attenuated. And breaking that down in more detail at the circuit level, the entorhinal cortex projects via the perforant path onto dentate gyrus granule cells, which then project forward to CA3. The perforant path also sends an excitatory projection onto parvalbumin expressing inhibitory interneurons in dentate, which inhibit granule cells. So this is a feed-forward inhibition. And then the granule cells also excite somatostatin expressing inhibitory interneurons, which then feed back and inhibit the granule cells. So there are feed-forward and feedback inhibitory loops that help to offset or balance the excitation onto granule cells. This balance of excitation and inhibition ensures that granule cell firing remains sparse despite this really strong excitatory input. But you can see that if something were to go awry, this could be a setup for runaway excitation. And in fact, the circuit has been the subject of a lot of attention within studies of adult onset or injury-acquired temporal lobe epilepsy, but it's not been studied in the context of genetic epilepsies or Dravet syndrome. So I'll now move on to the data portion of the talk, in which we begin by characterizing the circuit in SCN1A mice. So basically asking the question, is this corticohippocampal circuit hyperexcitable in the SCN1A mice? And my experimental setup was as follows. So I electrically stimulate the perforant path and then use 2-photon calcium imaging to record the resulting activation of granule cells within the dentate gyrus. And again, the major advantage of 2-photon calcium imaging is that we can record from dozens or even over 100 cells in an imaging field, instead of electrophysiology, where we would be sampling cells one at a time. This is an example of what a slice looks like. So you can see the stimulation electrode coming in to activate the perforant path, and then the imaging field centered on the dentate gyrus. And if you zoom in, this is what we see under 2-photon imaging. These mice are injected with a virus encoding the calcium indicator GCAMP, and you can see GCAMP really beautifully expresses throughout the dentate gyrus granule cell layer. And I told you in the introduction that calcium imaging provides an optical readout of action potentials, but we wanted to prove that that was the case reliably within our own experimental paradigm. So to do this, I performed simultaneous cell-attached recordings and calcium imaging under 2-photon of individual neurons. So shown here are example traces from one neuron with action potentials on the left and the delta F over F naught calcium signal on the right. And so you can see the bottom trace in blue. This is a subthreshold stimulation of the perforant path. So there is no action potential triggered and no calcium transient. And with a larger stimulation, I elicited a single action potential, and the GCAMP we're using, GCAMP 7S, is sensitive enough to detect even that one action potential. And you can see that the size of the calcium transient then scales with the number of action potentials. And looking at the data over multiple cells that we recorded from, you can see that nice relationship between number of action potentials and the size of the calcium transient. And importantly, that relationship is the same between the wild type and the SCN1A mice. And this was critical because it means that we can compare the results from our calcium imaging experiments, and we know that it's an apples to apples comparison in terms of what that tells us about ground truth action potentials fired by the cells. So let me show you what this data actually looks like. So this is a movie that will show you one of these slices, and you'll see the dentate gyrus that's quiet at rest, and then in response to perforant path stimulation, you should see many cells in the granule cell layer light up. And in case anyone was blinking just when it flashed, I'll play it one more time. So that's what our raw data looks like. So how can we take this visual data and capture or quantify it so that we can analyze it? So I'll take you through this graph in some detail, and then on the next slide, I'll show you multiple graphs that are with a similar format. So for each imaging field, I stimulate the perforant path across a range of current intensities, and that's plotted on the x-axis. And then after each, in response to each stimulation, we measure the proportion of granule cells that are activated by that stimulation on the y-axis. And in this graph, the dots are the data from individual fields at each current intensity. And then the light lines are the predicted binomial fits for each field across the range of stimulation. The dark lines are the average of the predicted fits, and the shaded area is the 95% confidence interval of the fits. And I'll point out that for these experiments, the question of what is the N for the experiment is actually quite a non-trivial question, because we have a certain number of mice, and each mouse can give rise to multiple slices. Within each slice, we have one or more imaging fields, and then within each imaging field, again, as a great advantage of 2-photon calcium imaging, we have dozens to over 100 cells. So this is a nested or hierarchical data structure, and in order to analyze this, taking account different levels of variability, or taking into account variability at each of those levels, we used a mixed model statistical approach. So you can focus for simplicity just on the dark lines, which are again the average of predicted fits. And I want to credit Dr. Alice Omaruthu in Dr. Ethan Goldberg's lab. She is a brilliant postdoc in the lab who spearheaded the really non-trivial analysis of all of this calcium imaging data. So now let me show you what our results are. So because the corticohippocampal circuit has been shown to be developmentally regulated, and because Dravet syndrome is a developmental disorder, we interrogated the circuit at 2 different time points. We tested early postnatal mice, P14 to 21, and young adult mice that are mostly in the P50 to 50s to 70s range. So these are mice in which the phenotype is just beginning to emerge, and then mice that could be considered to be in the chronic stage of the disease. And so shown here are the proportion of all granule cells activated in response to either a single pulse or 4 pulses of perforant path stimulation. And let me walk you through what our main conclusions are from this experiment. So first, you can see if you just look at the early postnatal time point, that there's a kind of trend toward increased activation in the SCN1A genotype, but it's a small difference and it's not statistically significant. On the other hand, in the young adult time point, you can see this wide gap opening up between the red and blue curves, in which the SCN1A circuit is much more excitable than in wild type. And then the other finding here that I want to draw your attention to is what's happening across development. So if you look at the wild type data, the blue curves, you can see that in the young adult time point, it's really difficult to activate this circuit, whereas it's activated much more readily in the early postnatal mice. And this is consistent with data that's been shown previously, looking at the way in which the circuit changes over time. So basically, there's a normal developmental regulation of the circuit in which it becomes much less excitable. So what we're essentially seeing is a replication of the literature in the wild type mice, but our SCN1A mice are seemingly failing to undergo that normal change in the circuit over time. And as I said, this is the proportion of all granule cells activated, but we were aware of the fact that within the cells that we're measuring, there might be essentially some false negative data. So there could be cells that we're including that are failing to be activated for non-physiologic reasons. So for instance, it may be that the perforant path projection onto those cells could have been severed by the slice preparation, or not targeted by the exact electrode placement. So we performed 2 additional types of analysis to account for that possibility. And I can go into this in more detail if there are questions, but I'll suffice to say here that our basically the same result that we found holds true across these different types of analysis. And finally, we analyzed not only the proportion of activated cells, but the amplitude of that response, shown here plotted on a log scale. So as I showed you, the magnitude of that calcium response is a proxy for the number of action potentials fired. So in other words, not only are more cells firing, but the cells that are firing more action potentials, especially in the young adult time point. So again, to recap our interpretation of these results, first, the dentate gyrus granule cells are overly activated by perforant path stimulation in young adult SCN1A mice. And this manifests both as more activated cells and as more action potentials fired in those cells that are activated. Secondly, this difference between genotypes really develops over time. The SCN1A mice appear to fail to undergo the large physiologic decrease in excitability, that's a normal feature of development. And whether this circuit deficit is indeed present early on, but it's masked by the relative hyperexcitability in wild type, or whether this circuit fails to mature from some combination of the genetic lesion or development within a brain undergoing seizures is not yet clear. And then I think it's really interesting that we find a similar circuit dysfunction here that has been described in animal models of injury acquired temporal lobe epilepsy, because these are epilepsies with totally different etiologies, but it seems like there's a convergence at this circuit level in the pathology in the brain. So the next question we sought to answer is, what is the mechanism of this circuit dysfunction? So what cell types or synapses give rise to this dysfunctional circuit? And my hypothesis going into this is that the parvalbumin expressing inhibitory interneurons would be the most likely culprit. So we know that PV cells and dentate really powerfully regulate the responsive granule cells to perforant path input. And as I mentioned, there's this kind of overarching interneuron hypothesis regarding the pathophysiology of Dravet syndrome and these SCN1A mice. So we hypothesized that the SCN1A PV cell dysfunction would be the culprit. And in order to test this, the first experiments we did were whole cell patch clamp recordings of the PV cells to test their intrinsic firing properties. And these experiments were done by Kevin Goff, who's a really talented MD PhD student in the Goldberg lab. And what we found are indeed some differences between genotypes, but they were more subtle than we'd anticipated. So I'll explain my interpretation of what we see here. So on the top, you can see example traces of wild type and SCN1A PV cells in response to a prolonged, large square pulse current injection. And you can see that in the wild type PV cell, the action potential height remains relatively constant across the pulse train. But in the SCN1A PV cell, there's this kind of gradual decline in the spike height. That difference that you can see by eye is captured in the graph on the bottom left, where we're quantifying the action potential peak across number of action potentials. And you can see it drops off faster in SCN1A. And as a sort of end consequence of this, by the time we challenge these cells with prolonged and increasingly large current steps, at some point the SCN1A cells fail to fire sooner than their wild type counterparts. So you can see that the early part of this input-output curve is indistinguishable between genotypes. The curves are right on top of each other. At around 2.5 times Rio-base, they start to diverge, because where the SCN1A firing fails, especially toward the end of a prolonged pulse train. And so again, there is a deficit here. It just seemed to us to be relatively subtle compared to the profound circuit dysfunction that that I just showed you. And we also thought probably in response to a single or even 4 pulses of perforant path input, effectively the PV cells would mostly be operating within a range in which there was not a significant difference between genotypes. So this difference really comes out under kind of extreme conditions that might not be where they're operating physiologically. So we kind of reconsidered the initial hypothesis and looked for potential other culprits. So the next question is, maybe it's the granule cells themselves. So maybe the granule cells themselves are hyperexcitable in SCN1A mice. So I tested that with a head-to-head comparison, and the take-home message, in short, was that there were no really significant differences. And then we finally asked, as I'd introduced, or as I'd reviewed in the introduction, the sparse activation of granule cells is because of this finely balanced excitatory and inhibitory input evoked by perforant path stimulation onto the granule cells, or perforant path input onto granule cells. So maybe that balance is off in the SCN1A mice. So I performed an experiment patching the granule cells, stimulating the perforant path, and then measuring the EPSC, which is a monosynaptic postsynaptic current, and the IPSC, which is disynaptic. And as you can see in these example traces here, the big difference that really actually stood out was that the EPSCs were a lot larger. So when we plot the E to I ratio across all of the cells. That ratio was a lot larger in the SCN1A granule cells, but it was really driven by an increase in excitation rather than impaired inhibition. And this was, to us, a surprising result because, as I've said, most of the SCN1A mouse literature has really focused on inhibitory interneuron dysfunction. And because this result was surprising, we wanted to do a sort of sanity check control experiment to test whether if we fix or reverse this more subtle dysfunction of the PV cells, would that be sufficient to fix the circuit? And we did this using HM1A, which is a spider-derived peptide toxin that's a selective activator of NaV1.1. And you can see, and this data was also collected by Kevin. You can see that when we add HM1A to the bath, it turns the SCN1A PV cells into cells that look more like the wild type. So you can see SCN1A on the left, there's that progressive decrease in spike height. And in the presence of HM1A, that same cell no longer has that decrease in spike height. And when it's stressed with large current injections, it takes a lot more to make that cell fail. And that's quantified at the population level in the graph on the right. However, when we added HM1A to the bath and performed that calcium imaging experiment with 2-photon imaging of granule cells in response to perforant path stimulation, there was no difference in the resulting fluorescence in the granule cells. So sort of as we suspected, fixing this more subtle deficit that comes out in the extreme activation of the PV cells was not enough to actually fix the circuit. So our conclusion was that by the young adult more chronic phase of the disease time point, the mechanism of corticohippocampal circuit dysfunction is more complex than just simply NaV1.1 insufficiency. So why does this matter? As a physician scientist in epilepsy, I think it actually matters a lot in terms of implications for treatments for Dravet syndrome and also the optimal stage of the disease at which to intervene clinically. So this was a really exciting preclinical paper that came out of Laurie Isom's group at Michigan, in which they used antisense oligonucleotides that increase SCN1A transcript levels within the mouse brain. And this worked really beautifully. It rescued the phenotype with decreased incidence of SUDEP and decreased seizure frequency. However, just to point out, they did this intervention at postnatal day 2 or 14. So these are itty bitty baby mice that have actually not yet developed the epilepsy phenotype. These mice start having seizures a little later than P14. So it might be that if you intervene early enough, just reversing the genetic lesion could be sufficient to rescue the phenotype. But maybe if you wait until the more chronic phase of the disease, like our young adult mice, you're contending not only with the initial genetic insult, but now with the many, many downstream consequences of that in terms of aberrant neuronal development and circuits not being what they're supposed to be. The next portion of my talk, I'll show you our data testing whether the circuit dysfunction could be implicated in actual seizure onset in SCN1A mice. And taking a step back, this is again where I'd like to convince you that the SCN1A mouse model is a really powerful experimental model to use. So in general, when we study seizures in animal models of epilepsy, you can evoke seizures chemically or electrically or otherwise. And this is experimentally advantageous because the seizure is predictable. You know when it will happen, so you can time your experiment accordingly. But it could be criticized as being unnatural, and maybe those seizures are in some key way not entirely mechanistically the same as a spontaneous seizure. On the other hand, you can study spontaneous seizures, but this is more experimentally challenging because the seizure can happen 2 a.m. on a Tuesday. So I think one thing that's very powerful about the SCN1A mouse model is by evoking seizures with hyperthermia, we have seizures that are, you could argue whether or not they're entirely natural, but I'd say they're at least naturalistic, especially given the fact that our patients with Dravet syndrome have seizures with fevers. But these seizures are also predictable. They happen within a few minutes, or you're able to predict their timing within a window of just a few minutes, so you can time your experiment. And I want to thank Nate Sotuyo, who's another graduate student in the Goldberg lab. And Nate and I worked together to basically optimize the protocols for evoking and reliably evoking and testing seizures in vivo in our SCN1A mice. So how can we use optogenetics? How can we add optogenetics to the mix to be able to manipulate hyperthermic seizures in our mice? So first we have to, again, introduce this tool, for instance, by injecting a virus into the brain, encoding one of these optogenetic proteins. And then we also implant in the brain a fiber optic cannula that delivers light to the region of interest. And this couples externally to a laser or a light source. We then photostimulate. This is a stock photo taken from the Dice-Roth lab showing blue light going down into the brain of a mouse. And then we can heat up our mice to evoke a seizure. And in our SCN1A mice, nearly 100% of them have seizures by the time their internal body temperature reaches 42.5. So in this experiment, I optogenetically activate the entorhinal cortex while coupling this with hyperthermia. So on the first day, in all of the mice, I elicit a hyperthermic seizure. This is to determine the seizure threshold for each mouse. On the second day, I return to the same cohort of mice and optogenetically stimulate first at room temperature, and then if no seizure is elicited, gradually elevate the body temperature of the mouse. And our major finding was that the stimulation was indeed ictogenic in the SCN1A mice. So we were able to trigger seizures at room temperature in around half of our mice. And overall, there was a decrease in seizure temperature threshold with optogenetic stimulation added. And in a separate cohort of wild-type control mice, we tested the same stimulation to ensure that we weren't just evoking seizures from an overly strong optogenetic stimulation. And none of these wild-type mice had seizures with stimulation at room temperature, and 1 out of our 5 mice had a seizure with stimulation coupled with heating. As a second control, we wanted to ensure that the decreased seizure threshold that we observed on day 2 was not just from a sort of kindling effect from these mice having had a seizure on day 1. So in a separate cohort of mice, we evoked hyperthermic seizures without optogenetic stimulation on consecutive days, 24 hours apart. And we showed we found no significant difference between their seizure temperature threshold. And then finally, I'll show you our attempt to rescue this hyperactivated circuit. And so as I showed you, we have data to support the idea that perforant path stimulation results in overexcitation of the granule cells, resulting from too much excitation, an imbalance of too much excitation, overwhelming inhibition. So our idea was to selectively activate the inhibitory component of the circuit to counterbalance that excess excitation. So again, I electrically stimulate the perforant path while performing 2-photon calcium imaging of the dentate gyrus granule cells. But this time, we add on optogenetic activation of the parvalbumin expressing inhibitory interneurons. So if we increase the inhibition, that should be able to cancel out the pathologically increased excitation. And here's that image that I told you would come back by the end of the talk. Those red cells are actually the PV expressing interneurons in the hippocampus in the dentate. And you can see how sparse they are. There are not a lot of them. So if we just tweak that particular part of the circuit, would that be enough to rescue this hyperexcitability? And this is also the promised combination of optogenetics and calcium imaging, as I said, we get to by the end of the talk. So these mice are double transgenic mice that are SCN1A mice in which the PV cells also express Cre recombinase. So we can inject these mice with a combination of 2 viruses, again, a virus encoding GCAMP expressing throughout the dentate gyrus, and then a second virus encoding a Cre-dependent crimson, which is a red-shifted excitatory optogenetic protein. And these have complementary spectra, so we can do a sort of all optical experiment with dialing in and reading out neuronal activity in the circuit. And shown here are traces from individual cells of the calcium signal, the calcium recording in response to electrical stimulation alone, which is shown in black, or electrical stimulation paired with simultaneous optogenetic activation of the PV interneurons, and that's in gray. And you can see that by bringing the PV cells more strongly online, it really kind of profoundly decreases the response of the granule cells. And that's shown at the population level here. So each line here is data from one imaging field. And you can see the proportion of activated neurons and then the mean magnitude of the activation in the cells that were activated. So to summarize my results, first, I've demonstrated that the corticohippocampal circuit is profoundly abnormal in young adult SCN1A mice. And again, this is a maybe convergent circuit dysfunction between different epilepsy types, because a similar finding has also been shown in models of acquired temporal lobe epilepsy. And second, this circuit abnormality worsens with or is unmasked developmentally over time. Third, this abnormality may be driven by increased excitation onto dentate granule cells in SCN1A mice, and it's not as simple as just insufficient NAV1.1 expression at this older time point. Fourth, optogenetically driving entorhinal cortex is ictogenic in SCN1A mice. And finally, targeted increase of PV cell firing is able to rescue the hyperactive responsive SCN1A granule cells to perforant path stimulation and slice. And then I'll end with, I have just one slide each, kind of at the for some high level thoughts about future directions. So first, short-term future directions, which is essentially my K08, which will be starting after I finish my clinical fellowship this year. I think the work that I've just shown here raises many questions that could be pursued in follow-up. So first question is, what is the source of this increased synaptic excitation onto the granule cells? So is this presynaptic and or postsynaptic phenomenon? And I think there are several interesting experiments that we could do to try to answer this question. Another direction is, so we demonstrated that driving entorhinal cortex was sufficient to stimulate seizures in these mice, but how about modulating excitability within the dentate so that the response to whatever cortical input is different. So we could, for example, using chemogenetics, tune the seizure threshold by modulating excitability of the PV cell or granule cell populations. And then what are the implications of this entorhinal cortex to dentate hyperexcitability? Does this excitability propagate forward to CA1? So for instance, could we image in vivo and see if there is a resulting downstream consequence in terms of pathologically hyperexcited CA1? And then longer term, again, this will just be sort of some high level thoughts. I think one thing that's really fascinating about epilepsy is that it's a disease that exists in 4 dimensions. What I mean by that is I've been showing you throughout this slide a circuit diagram that looks something like this. So here it's in 2 dimensions, but of course in the brain it's 3-dimensional. But I think it's actually a little misleading to show this because what's really challenging and fascinating clinically in epilepsy is that seizures are discrete events. So you can have a patient in your clinic who has a completely normal neurologic exam, and they're essentially telling you the history about something that happened a week ago. And it can be completely true, even if you don't see it with your eyes at a different moment in time. And even someone with really terrible epilepsy is typically only actually having seizures just a fraction of the time. So this is challenging and interesting clinically. And I think on the research side, there's a sort of related challenge, which is how can we actually know or be sure that the circuits that we're studying are the dynamic circuit dysfunction that would really give rise to seizures, or the transition in the brain between the interictal and ictal state, as opposed to chronic deficits associated with an epilepsy phenotype. So for instance, the corticohippocampal circuit dysfunction that I was showing you here is presumably present in a static way, but why does the brain, these mice are not having seizures all the time? What is it that's changing as they flip between the not seizure and seizure state? So I think it will be really interesting in the future to move in the direction of asking what changes are seen, for instance, in this corticohippocampal circuit during the state transition and the time of seizure onset. And I mean, this really relates to the fundamental question in epilepsy of when and why do seizures happen. So, when is it? What is it that makes a seizure happen at that time? And we would love to know that question. We'd love to know the answer to that question clinically for sure. Within the SCN1A mouse model, this can be a slightly narrower question because we again know that these seizures can be triggered reliably by hyperthermia. But even the answer to this question we don't know, and I think is a really interesting one. So what is it that's happening in the brain of these mice as their body temperature elevates that then interfaces with these canonical corticohippocampal circuits to switch to this ictal state? So that basically concludes my talk, and I want to thank the many people who mentored and collaborated with me in this work, especially, again, first and foremost, Dr. Ethan Goldberg, who's a fantastic physician scientist and mentor, and also my funding sources. And thank you again for the invitation, and I look forward to discussion.
Thanks, Joanna. That was great. That was a ton of work. You did a great job with that. So we have time for questions. If anybody has a question. Momoko has a question. Let's see. She says, is there any difference in the number of PV somatostatin cells between the SCN1A and control mice? And is the hippocampal anatomy normal in these mice? By the way, great talk.
Oh, thanks. Okay, so those are really good questions. I'll say within our, in our lab, in our mice, we have not done that experiment for like count and quantify PV or SST cells. I think in general in these mice, the inhibitory dysfunction is not thought to be from a loss of cells, but more just a change in their intrinsic properties. I think, so there is some data, not from our lab, that there may be some difference in the hippocampal structure. So there's nothing as simple as, like, these mice don't have like an overt MTS, mesial temporal sclerosis. There was a paper in which they looked at the branching structure of granule cells and how that changed over development, and there were abnormalities. So it may be that some more sophisticated immunostudies would be able to detect differences in cell number or number of branches of the projections or synapse count, like that. But those are not experiments that we've done.
So I've got a question. So in the mice, it seems pretty clear that SCN1A is primarily expressed in the inner neurons, not just PV, but a couple of the other types. I mean, not so much in the excitatory cells, but in the human literature, especially from the stem cell sort of debate, there's a debate, right, between and the SCN1 gene is probably the most. This debate's the most robust. Like, is it mouse specific or are human cells different? And so what do you think might be going on here, with that, and how would that influence all this work that you've done in the mouse?
Yeah, so I will say there's one particularly for us notable exception to the rule. So as you said, in the mouse, most of the NAV1.1 expression seems to be in inhibitory neurons. One maybe very interesting exception to that rule is NAV1.1 is expressed in a subset of entorhinal cortical excitatory cells that do project to dentate. So we're very interested, relevant to this work, we're really interested in going after that specific subset of cells.
You know which ones?
Sorry?
Do you know which ones?
Oh, I don't know how to answer that question. I mean, they're excitatory cells in entorhinal cortex that project via the perforant path.
Is it the medial or lateral? Is it what layer?
I think that they're layer 2.
Yeah, and they're projecting right to the dentate. There's different types out there. Yeah.
Okay. Yeah, I think there's a rich literature that I don't know well enough yet.
Oh, that's really interesting. Yeah. But the granule cells themselves don't have SCN1A.
No.
Okay. So we have another question. So I could ask you lots of questions, but we'll go on. So we have great talk, Joanna. Do you know if there is any change in role in the Mossy cells in the dentate?
Oh, okay. The short answer is that I don't know. It's possible that there is a paper that I'm forgetting that would answer that question, but it's not in my head. And it's not something that we've directly investigated.
Any more questions? Going once, twice. This is a great talk. You guys obviously have a lot of work. And you said this paper is coming along, hopefully.
Yes. So
I'll put pressure on you.
yeah, I mean, there's always the question of there are always more experiments that we could do
Yeah.
that I think would enrich and strengthen the story. So there's always a question of when do you stop? But I think something that roughly looks like the talk that I just gave should be available at least on bioarchives in the relatively short term.
That's great. Yeah, this is great. And thank you so much for giving your talk. I think this is a great seminar, and we really appreciate you coming. And thank you everybody for attending.
Yeah, thanks so much for the invitation.
Okay.